Now we will discuss one of the analytical procedures in the dairy industry, which is detection of protease in milk. As the name suggests, that protease is an enzyme that acts on proteins. It is available in the milk from different sources. They might be indigenous sources, like it comes from the milking animal, or it might be the exogenous resources, such as it might come from microorganisms that are present in the milk, and those microbes could have entry into the milk from all the various routes, as we discussed. So it has got really important application in the dairy industry. We need to know the protease enzymes, their presence and their activity in the milk, in order to know the final product quality, because this protease can produce certain undesirable changes in milk, such as unpleasant flavors and odors in milk. So there are a lot of different kinds of bacteria which are available for these protease enzymes, because this is a normal process for bacterial growth, that they would be breaking down the proteins present in milk, so they would need these enzymes. So one of the major bacteria that is involved for these proteolysis reactions in milk is Bacillus subtilis. It's a pathogenic bacteria. It's a psychotroph. As I said, that the psychotroph bacteria can grow at lower temperature. These microbes can also produce some of the hetero-resistant proteases that can stay in the milk, and even the pasteurized milk or sterilized milk still can have some of these proteins, the protein breaking enzymes. So that would mean that your final product might degrade very quickly if they have got these proteases enzymes. So that is why we need to know the activity of proteases in milk. So there are different methods available for determination of this protease activity, but we'll discuss one of them. So we need to prepare the sample first. It would depend upon the type of method that we are going to use here, and also the type of milk that we want to analyze. Normally we would need some standard protease enzymes. Normally we can take, for example, here Bacillus subtilis protease enzyme that is present commercially, that has to be taken as a standard here. If we talk about the reagents used in this process, there are three different reagents. We use disodium hydrogen phosphate and sodium dihydrogen phosphate and trichloroacetic acid. These are three reagents that are used. So as a procedure, as a first step, we would produce a buffer solution of pH 7.2, and for making this buffer solution we actually add up these two reagents, as we discussed. 36 milliliter of sodium hydrogen phosphate, that is 5 millimolar concentration, and 14 milliliter of sodium dihydrogen phosphate, again 5 millimolar together. So that would make up 50 ml of the volume. Then we dilute this 50 ml to 1000 milliliter, that is 1 liter. So that reagent or this final stock solution would be used for further analytical procedures. Then this buffer is used to prepare 1% azocasein solution. This is again to be used for further process in this analytical procedures. Then we prepare another solution, which is 5% aqueous solution of trichloroacetic acid. So that would be needed for the next process. Now let's see how we actually determine this protease activity in milk. So we add 1 milliliter of azocasein solution with 100 milliliter of sample, which is milk or any of the dairy product that we want to measure the enzyme activity. Then these contents are mixed together and incubated at 35.5 degrees C in an oven for 15 minutes. So once this reaction has taken place, the enzyme have actually worked with all the proteins present in the sample. We would add this 5% trichloroacetic acid solution, 2 milliliter of that. So this addition would actually stop this reaction of enzyme with the protein. Now the last step we would be having a supernatant fluid that would be determined by spectrophotometer, UV spectrophotometer. So this spectrophotometer would be measuring the absorbance at a wavelength of 345 nanometer. So once we know this absorption, this is then again compared with the standard solution of proteases at different concentrations. So this standard curve would actually give us the final protease activity of this milk in our sample that has to be tested here. So this was all one of the simple procedures for measuring the protease activity. And I would also again say that there are more than one methods, but this method is generally used in the industry for protease determination.